Welcome back to the Scilife Academy. In this short course, we will examine 5 types of sterilization methods used in life sciences. We'll take a look at heat sterilization, wet and dry, radiation sterilization, gamma radiation and UV light, and electron beam sterilization. Filtration sterilization and chemical sterilization will be looked at in another session. Let's start with the different ways of categorizing sterilization methods. In a nutshell, sterilization can be categorized into 4 types: heat, gas, radiation, and filtration. Another way to classify sterilization methods is by physical and chemical sterilization methods. Physical sterilization methods involve the use of external energy sources to destroy microorganisms. These include heat sterilization, radiation sterilization, and filtration. The choice depends on the capacity of the formulation and the package to resist the treatment applied by the sterility procedure that is selected. Let's have a look at each one. Sterilization items are either heated wet or dried. Wet heat sterilization is also known as steam sterilization, steam under pressure sterilization, or autoclaving. For example, rubber and durable plastic materials can be sterilized by wet heat in an autoclave, which uses steam under pressure. Moist involves raising the temperature between 121 and 134 degrees Celsius, with a pressure above 15 psi or 0.5 bar. At this pressure and temperature, water becomes saturated steam in an autoclave. These conditions denaturalize cell proteins and DNA of microorganisms, with a sterilization cycle between 10 and 60 minutes. One advantage of using steam sterilization is that it can easily penetrate the inside of medical supplies. At 170 degrees to 180 degrees Celsius, dry heat sterilization destroys microorganisms by oxidation. Heat coagulates proteins, causing the cells to burn and be exploited. Dry heat is effective for a variety of materials such as glassware, glass containers, stainless steel equipment, oils, and dry powders, provided the powders are capable of withstanding high temperatures. Dry heating sterilization is especially effective at 250 degrees Celsius at destroying endotoxins. Is there another benefit to using dry heat? Sure, it keeps materials dry post-sterilization, which prevents corrosion. Is there a disadvantage to dry heat? Unfortunately, yes. The process with dry heat is very slow and difficult to control. In order to kill resistant spores, longer exposure times are necessary. It is 10 times less efficient than steam sterilization. Which is better, dry heat or wet heat? Wet heat with moisture is more effective.